Hello to the ladies and gentlemen who are watching this video right now. We, Lay, Parvin and I, Ryishi, are going to educate you guys about this topic, which is metabolic processes. Okay, Lay and I will be covering energy and cellular respiration part in this section, whereas Parvin will be taking care of the photosynthesis part of this section. So, let's go. But wait. Let's change the music first. It's a bit too dramatic. So, okay, let's go. What is metabolism? It is the chemical reactions that change and transform matter and energy in cells. It can be split into two different types of metabolic pathways, which are anabolic pathway, which is taking in energy to make the smaller molecules bigger, and catabolic pathway, which is the release of energy in the process of making bigger molecules smaller. Energy plays a major role in these reactions in order to get the products wanted, which leads us to thermodynamics. It can be split into two different laws. Our first law is energy can never be created or destroyed. It can only be transferred. In the second law of thermodynamics, in any process, the universe tends to disorder, and in a special known term, they call it as entropy. In all types of reactions, energy is either being used or released, and it is known in two different kind of terms. The terms are endergonic and exergonic. Endergonic reactions are reactions that require energy. On the other hand, exergonic reactions are reactions that release energy. ATP is directly involved in the process of metabolism of either its usage or the synthesis. And ATP is the source of energy in the reaction glycolysis. But what is glycolysis? Glycolysis involves in breaking down glucose into smaller molecules in the form of pyruvates, and as a result we would actually get two of those pyruvates. And of course, ATP plays an important role in this process. In the first step in glycolysis is the conversion of glucose into glucose 6-phosphate. The enzyme that catalyzes this reaction is hexokinase. The second reaction of glycolysis is the rearrangement of glucose 6-phosphate into fructose 6-phosphate by glucose phosphate isomerase. Phosphofructokinase changes fructose 6-phosphate into fructose 1,6-biphosphate. The enzyme aldolase splits fructose 1,6-biphosphate into two sugars that are isomers of each other. These two sugars are dihydroacetone phosphate and G3P. The enzyme triphosphate isomerase converts the molecules dihydroacetone phosphate and G3P. Phosphoglycerate kinase transfers a phosphate group from 1,3-biphosphoglycerate to ADP to form ATP and 3-phosphoglycerate. The enzyme enolase removes a molecule of water from 2-phosphoglycerate to form phosphoenol pyruvic acid. The enzyme pyruvate kinase transfers a P from phosphoenol pyruvate to ADP to form pyruvate acid and ATP. Okay, now let's go to the rest of this section of energy and metabolic processes. And Lay will be explaining the remaining parts of this section. Let's go.
First, the pyruvate will comes in, and then the CoA, which is the coenzyme A, will diffuse into the reaction. NAD will reduce to NADH, which gain one hydrogen electron with the enzyme of dehydrogenase. The carbon dioxide also will diffuse out, so the reduction occurs in this reaction. In this stage, acetyl coenzyme A is formed and binds with the oxaloxytate, which is the 4 carbons compound, and then the acetyl coenzyme will produce the citrate by the citrate synthase. The water also will diffuse in, which is hydrolysis. CoA also will diffuse out. Citrate is a 6 carbon compound and produce isocitrate by the aconitase. Isocitrate also a 6 carbon compound. Before proceed to the next stage, the carbon dioxide will diffuse out, so will lose 1 electron, so the oxidation is occur in this stage. NADH plus will become NAD by the isocitrate dehydrogenase. The oxidation is occur in this stage. Next is A-ketoglutarate. Before proceed to the next stage, the CoA will diffuse in this reaction, and then the carbon dioxide will diffuse out in this reaction. So the A-ketoglutarate will lose 1 hydrogen electron, so the oxidation is occur. The NAD will become NADH plus by the A-ketoglutarate dehydrogenase enzyme to form succinyl-CoA. In this stage, succinyl-CoA only remains 4 carbons. HS-CoA will diffuse out. GTP plus pi will convert to GTP. In this reaction, ADP also will convert to ADP, which is an energy, by the enzyme of succinyl-CoA synthase.
Succinate is formed. The FAD will become FADH2 by the succinate dehydrogenase and form the next stage of fumarate. The fumarate will proceed to the next stage to the mellet by the hydrolysis and also by the fumarase enzyme. Mellet proceed to the oxaloxytate. The NAD will oxidize to NADH, so the oxidation is occurred in this stage by the mellet dehydrogenase.
And now we look at photosynthesis. Photosynthesis, the energy of life. We're first going to take a look at the structure of the organelle responsible for photosynthesis, chloroplast. This is the structure of a chloroplast. Now let's move on to photosynthesis. Photosynthesis is divided into 2 stages. The first stage being the light-dependent reaction, in which water molecules are split as light energy is absorbed and transformed into chemical energy in ATP and NADH. We now move on to the second stage, which is known as the Calvin cycle, also known as light-independent reaction. In this, carbon dioxide is converted into sugar with the use of ATP and NADPH. We now move to photosystem 2, where splitting of water molecules into electrons, protons, and oxygen gas occurs. This is then followed by the infamous electron transport chain, which starts with the oxidation of P680. This is followed by the oxidation reduction of plastoquinone. Then, electron transfers from cytochrome complex and shuttling by plastocyanin. Oxidation reduction of P700. Electron transfer to NAD plus by ferredoxin, NADP plus. And last but not least, the formation of NADPH. This is soon followed by ATP synthesis
where ATP is synthesized as protein moves through the ATP synthase complex. We now move on to Calvin cycle itself. Calvin cycle is divided into 3 phases. The first phase being carbon fixation. In carbon fixation is where the conversion of carbon from an inorganic to an organic form. We then move on to the next phase, which is reduction, where molecules of 3-phosphoglycerate gets an additional phosphate added to from hydrolysis of ATP. We move on to the final stage, which is regeneration, where some of the G3P molecules are combined and rearranged to generate RePB that is required to start the cycle over again. Alright, now for alternative methods of carbon fixation. To start things off, we can look at the prevention of water loss. Now this can be done through the stomata of the leaves. Simply controlling the opening and closing of the stomata according to the time of the day can prevent water loss. We now move on to C4 plants. More specifically, C4 cycle. Now, the C4 cycle is an alternative form of carbon fixation that some plants use in particularly hot weather to increase concentration of CO2 available. And now we look at CAM plants. CAM plants are a metabolic pathway used by mostly succulent plants in which Calvin cycle and C4 cycle are separated in time for better efficiency of CO2 fixation. Now we move on to the best part of it all, magical unicorns. Nah, just kidding. Experiment. So we decided to do an experiment on metabolic rate. Now we could have gone for the easy, boring yeast experiment, but we decided to study the effects of a human body towards the change in their surrounding. And our hypothesis was that when a human test subject is subjected to an extreme change in temperature, the blood glucose level of the human test subject will decrease. So then we did the cruel thing. We sent our human test subject to frozen? No? Well, a cold place and a hot place. And this is pretty much how we carried out our experiment. Alright. And here is our results from the experiment. The results seem very promising, where blood glucose level took the biggest dip while the other measured variables had very significant changes. And now this abruptly jumps to our conclusion, where we believe that our hypothesis is accepted and the human body did show a drop in glucose level, showing that cellular respiration did occur to constantly provide energy to every cell to regulate their internal temperature. And that about sums it up. Thank you for listening. The end.